Hello everybody and welcome to our cell cycle and cell division lecture. Today we'll be talking about the cell cycle, which is a predetermined sequence of events that in a cell's life that leads up to cell division. And cell division, of course, is the process of making new cells. All right, so only cells that are going to divide go through the cell cycle, and the end result of the cell cycle is the creation of new cells, but more stuff goes on in the cell cycle other than just creating new cells. There are two types of cell cycles because there are two types of cell division, so whichever type of cell division is happening, that's what kind of cell cycle is also happening. And one thing to keep in mind is that the cell cycle is very tightly controlled by the cell, and that is so that cell division, or the creation of new cells, does not occur when it's not supposed to. All right, so cell division is the process to make new cells. All cells come from pre-existing cells. That's something called the cell theory. A theory, by the way, is something in science that is well-known and has a well-known phenomena and has a lot of scientific evidence behind it. All right, two types of cell division are mitosis and meiosis. We are first going to generally talk about each of these, then we're going to go through mitosis in detail, and then we'll go through meiosis in detail. And then at the very end, we'll talk just a little bit about why these things are interesting. All right, so mitosis is used to make exact copies of pre-existing cells. So when I mean exact, I mean that the DNA base sequence is the same in the new cells as in the starting cell. By the way, the starting cell is called the parent cell, and the new cells that are made are called the daughter cells. Okay. We use mitosis to create new cells for growth. So all of us started out as a fertilized egg. This is also called a zygote. So you'll see that on some slides and in some activities or labs that we do in class. A zygote or fertilized egg is created through the union of an egg and a sperm. Okay. Once you have that fertilized egg, it needs to make new cells in order to get bigger, and the way it does that is through mitosis. So first of all, you start with one single cell. It divides by mitosis enough to make a baby, and divides some more to make a child, divides some more to finally make the adult. Once we're adults, we don't divide so much by our cells don't divide so much by mitosis to grow as to repair. We also see this process in plants. So this is an onion root tip. It's like at the very tip of the root where growth is happening, and it's in this region here. You can see cells in different parts of mitosis, and that's where the root is getting longer. Excuse me. All right. We also use mitosis to create new cells for repair and replacement. So if you injure yourself and you need to grow new cells to repair that injury, then that's through mitosis. We also are always doing mitosis in some parts of our body where new cells are constantly being made. For example, our skin cells are constantly being regenerated as they are sloughed off, and so we have constant mitosis happening under those top layers of our skin. And then finally, some organisms use mitosis for asexual reproduction. Asexual reproduction is really the process of making clones or exact copies. So bacteria do this, and then also some other more complex organisms. This guy here is called a planaria. He can regenerate the entire body by just a piece, and that creation of new cells is done through mitosis. There are other organisms that do this that you might be familiar with, such as a starfish. You can cut up starfish into a bunch of pieces, and each piece will grow a new starfish. All right, and then there's meiosis as the second type of cell division. Meiosis is a very special type of cell division that makes eggs or sperm, which are called gametes. So women make eggs, and they only make eggs in the ovaries, and men make sperm, and sperm are only made in the testes. Okay? So these would be and not just humans, it happens in any organism that does sexual reproduction. All right, so we're going to move into talking about mitosis, the mitotic cell cycle, and the process of mitosis. And I know this is such an exciting topic. Yay! Okay, so cell cycle describes the events in the life of a cell that is actively dividing by mitosis, and there are two parts to the cell cycle. There's interphase, and this is actually where the cell spends most of its time just being a cell, and then the other part is the actual cell division, which is actually composed of mitosis and cytokinesis. Okay, so here is a picture of the cell cycle. So interphase, we're going to talk about interphase right now. Interphase is basically everything excluding mitosis and cytokinesis. So I'll try to circle it here. So the G1 phase, the S phase, and the G2 phase, all of that is interphase. Okay. The G1 phase, also called the gap 1 phase, is where cell growth happens. So cells, when they are just made, are small, and so they need to get bigger. It is also where the cell just does its cell stuff, right? So making proteins, transporting things, etc., just doing its thing. Okay. Then there is the S phase. The S phase stands for synthesis, the S does, and that is also known as DNA replication. So synthesis for synthesizing DNA. DNA replication is essential for making two cells. So you're going to start with one cell that has one set of its chromosomes, or its DNA. It needs to copy its DNA so that it has enough DNA to go into both cells that are going to be created. And then there is the gap 2 phase, and this is further preparation for cell division. In this stage is really where a lot of regulation happens. And in a cells obviously can't ask themselves questions, but in a using proteins and pathways, the cell is basically asking itself, did I replicate my DNA and did it replicate correctly? If yes, go ahead and divide. If not, stop, and maybe even be destroyed, or maybe just stop where you are. Do I have enough organelles for two cells? Do I have enough building blocks like amino acids, nucleic acids, nucleotides, carbohydrates, etc., in order to fund the growth of two cells? If the cell does not, then it would stop. So this is where a lot of regulation happens. Okay, and then you see this guy up here called the GO phase, or the G0 phase, I guess. The G0 phase are for cells that are just created, and then they aren't ever going to divide, so they just hang out in basically the G1 phase. It's essentially the same as G1. Normal ops, cell just being a cell, and it's never going to divide, so it stays like that. Many of our cells are like this, unless they're injured. Our muscle cells, for example, our brain cells, for example, those cells are just, once they're made, they're just being cells, and they won't divide. Okay, let's focus a little bit on the DNA replication, or the synthesis phase, the S phase. Like I said, it's necessary so that the daughter cells both have DNA. Okay. DNA polymerase is the enzyme that makes DNA, okay, and it's a cousin to RNA polymerase, which hopefully sounds familiar to you from when we talked about gene expression. So RNA polymerase is the enzyme that makes RNA. DNA polymerase is the enzyme that makes DNA. All right, and here comes the terminology. So there's lots of terminology with learning about cell division. Once the chromosome is replicated, it then consists of two sister chromatids. Okay? The sister chromatids are identical because they were made through DNA replication. So that means, let's see if I have a slide. No, go back for a second. So that means that the base sequence in the original chromosome is also the base sequence of both of the sister chromatids. Okay? They stay stuck together at something called the centromere. It's a complex of proteins that holds them together, and they're going to stay attached at the centromere until they're separated by cell division. Why might they be stuck together until they're separated? And so the cell can organize them. During cell division, these two sister chromatids need to be separated, and one needs to go to each new daughter cell. So why not keep them separated and keep them organized and paired up until it's time to separate them? All right, so now we're going to go through mitotic cell division. There's really two components, mitosis and cytokinesis, although they do bleed into each other. So mitosis technically is the division of the nucleus, where the parent cell's replicated DNA is going to be distributed equally among two daughter cells, and between two nuclei. And then cytokinesis is the actual division of the cytoplasm. Mitosis starts, and then at the very end, cytokinesis kicks in concurrently with mitosis. All right, so 4 phases of mitosis are prophase, metaphase, anaphase, telophase, and cytokinesis. I put those two together because they happen simultaneously. It's good to keep this in order, prophase, metaphase, anaphase, telophase, cytokinesis, because we'll see the same phases only named slightly differently in meiosis. So you might as well memorize it. Here is one way to memorizing it. Procrastinate more at the coast. You can make up your own. I suggest making up one that makes sense for you or that you think is funny. It will help you remember it. Here is a video that you need to watch. So watch this video of mitosis. If you haven't watched it yet, you could pause it and watch it now. Then I'm going to go through all the parts of mitosis in this lecture, or you could watch it after you finish the lecture. Maybe even consider watching it a couple times. Mitosis is a dynamic process. Once it starts, it just goes through the process and finishes it. We're going to look at it, phase by phase, as if these are, processes that happen, then stop, then restart, when it doesn't happen that way. So the video is nice for that. I couldn't get it to play the sound within the lecture, so I'm just keeping it separately, but make sure you watch it. All right, so we are going to start out. So we're going to go through all the phases of mitosis and what happens in them, okay? We are starting out with a parent cell that's in interphase, okay? This would be in what phase of interphase? It would be in G1. How do I know that? Well, its DNA is not replicated. I don't see any X's. The X's indicate the sister chromatids, okay? So that cell is going to need to go through S phase. It will also need to go through G2 phase and pass all of its checkpoints to make sure it's ready to go. Okay. When it does, then it will enter prophase. Prophase is the first part of mitosis. Okay. In prophase, the chromosomes condense, and when you're looking at a real cell, in interphase, the cell is just a squiggly mess, or the nucleus is a squiggly mess of chromosomes. You can't see individual ones. But as the cell moves into prophase, the chromosomes start to condense. They get more and more highly packaged, and you can see them as individual chromosomes. They look kind of like big fat worms or something like that. They are condensed so that they can be moved around easily and in an organized manner. Probably the biggest challenge that cells have during division is keeping that DNA organized to make sure it gets distributed to the daughter cells properly. And then the other thing is that the nuclear envelope will break down, and that's why this is shown here with a dotted line. Okay, the nuclear envelope breaks down, and all the organelles and stuff are just squeezed to the side of the cell, and the whole main part of the cell is given over to the process of dividing that DNA between the two new cells. And then finally, the last thing that happens are these things called mitotic spindles. Grow out and attach to the centromere of the chromosomes. The spindle fibers are part of the cytoskeleton, and they are really what control the chromosomes. And remember, each chromosome consists of 2 sister chromatids. You often see it as an X. It's one sister chromatid. Here's another sister chromatid. Don't know why it did that. There we go. So the mitotic spindles, or the spindle fibers, will control moving those as the cell moves into metaphase. Okay, so the spindle fibers are responsible for moving all of these chromosomes to the middle of the cell. Okay, when that happens, you know that you are in metaphase. Okay, so mitotic spindles, or I keep saying spindle fibers, that's a fine way to say it too, align the chromosomes and the metaphase plate. The metaphase plate is not a real thing. It's just the center of the cell. Okay, so there's the metaphase plate. You could also draw it any orientation, it will be the same. That's why I put this here. These two things are equal. They've just been rotated, but they show the same thing. Okay, and you can draw the mitotic spindle in there if you wish. Okay, so metaphase is just aligning those chromosomes at the center of the cell. It's really getting ready to then pull the sister chromatids apart and distribute those chromosomes to separate cells. Okay. Next, we're going to move into anaphase. Anaphase starts when the spindle fibers pull those sister chromatids apart. Okay, so it's pretty interesting with the terminology. It gets kind of confusing because hold on, let me finish drawing these. When we are here, we call each one of these an individual chromosome. So there are 4 chromosomes, even though they're replicated. So we would say that there are 4 chromosomes, each consisting of 2 sister chromatids. Here in anaphase, as soon as they start getting pulled apart, we now get to refer to each sister chromatid as an individual chromosome because they are going to be headed to their own new cell. Okay. So anaphase really starts when the chromosomes, sister chromatids, start being pulled apart, and it's going to last all the way until those sister chromatids reach the opposite poles. So they're going to be pulled in this direction towards the pole, and these ones will be pulled in this direction towards the other pole. Not really great arrows, but hopefully you get the gist. Okay, and like I said, they become individual chromosomes now. Okay. Once they reach the pole, you have reached telophase. Okay. In telophase, the new chromosomes have reached their poles and the nuclear envelope forms, just like it's doing the opposite of what it did in prophase, creating 2 nuclei, and cytokinesis is going to begin. This right here is called a cleavage furrow. That's how animal cells do it. It sort of like pinches it off into 2. Plant cells do it a little differently. They have to build a cell wall, and they start building the cell wall on the inside. And then cytokinesis is going to divide that in half and divide the membrane and the cytoplasm into 2 cells. Okay. And then voila, at the end of mitosis, you end up with 2 daughter cells that are genetically identical to each other and to the parent cell. Okay, so that means the sequence of bases that the parent cell had is the same as the 2 daughter cells. Okay, and so that's the end of mitosis. Might not be a bad idea to pause it now and watch that video before we move into meiosis. All right, so here comes up meiosis. Meiosis is an entirely different type of cell division, although there are some major things that are the same. Okay, so we're going to go through the meiotic cell cycle and then the process of meiosis, and I know that y'all are very excited, yippee, for this. Actually, you really should be excited because who is not interested in sex, right? And really meiosis is all about this sexual reproduction. That's why we have meiosis. Okay, so here's the meiotic cell cycle. It looks quite different than the cell cycle for mitosis. Okay, it's has a starting point and an ending point rather than being circular. That is to say that a cell will start the process of meiosis and then end it as 4 cells, and there's no regenerating one cell that's going to go through meiosis again. You just get the end point. Okay, the cell starts in interphase. The interphase is exactly like the interphase of mitosis. So there's a G1 phase, an S phase for DNA replication. The G2 phase also has regulation at it to make sure that it's all ready, that the cell is ready to divide. Okay. And then the cell is going to divide by meiosis, and the end result is not 2 cells but 4 cells. Okay, and these are the gametes. What are gametes, you may ask? Well, gametes are the eggs or sperm that are necessary for sexual reproduction. So we could call them sex cells also. Sometimes people call them that. Okay, and what happens is you start out with a regular cell, and then it goes through the process of meiosis and either creates an egg if you're a female or a sperm if you're a male. Okay, and those are then they fertilize, the sperm fertilizes the egg, and then you get the zygote and then the baby, and that's the sexual life cycle. Okay, the sexual life cycle is actually beneficial from a evolutionary point of view because it creates more diversity in the gene pool, because you're not just replicating the DNA you already have into a new organism, you're combining and creating new combinations, and that gives is more likely that some new combination will be able to withstand changes in the environment or other such things, and that the whole species as a whole will continue. We can talk about that more when we get to evolution as well. All right, before we go through the process of meiosis, there is more terminology. Okay, you already learned about sister chromatids. Those are the two identical chromatids produced by DNA replication. Okay, so there is one, I'm going to circle the blue one chromosome consisting of two sister chromatids. Okay, and you would have the same for each one of those. Okay, but we have more terminology. We also have something called homologous chromosomes, also sometimes called homologs, and this is a pair of matched chromosomes. They have the same size and the same shape as each other, and probably more importantly, they carry genes controlling the same inherited traits. So if you have a trait for hemoglobin on one gene, on one of your homologs, you will also have the gene for hemoglobin on your other homolog. That is to say that they carry genes controlling the same inherited traits, but they don't necessarily have the same versions of those genes. Different versions are called alleles. That's really a preview to when we talk about genetics, and we'll definitely talk about that more then. Right now we're just going to focus on what the heck are these homologous chromosomes. A couple other things about homologous chromosomes is that genes are located in the same place. Not only do they carry the same genes, but the genes are located in the same place. And so each one of us, as a human being, and many other organisms as well, have homologous chromosomes, and you get one from your dad and you get the other from your mom. So half of your pair is from dad and half is from mom. And how did that come to be? Well, backing up here, that came from the egg, okay, and from the sperm, right? So we get our what are called maternal homologs from mom and our paternal homologs from dad. All right, so let's look at a simple example here. Okay, so here is one chromosome with looking at only one gene, okay? There are, of course, many other genes on each one of these chromosomes, but we'll just look at one, okay? We're also beginning here to start using letters to represent the different genes, and different uppercase or lowercase to represent the different alleles. This again is also a preview to talking about genetics. Okay, so say here we're looking at a gene that codes for an enzyme that makes melanin. Okay, and there's actually two versions. Either you can make normal melanin or you might make a defective one. Okay. At any rate, focusing on the homologous chromosomes, here you have, say, this is the paternal chromosome. That meant it came from the sperm when this person was originally created, right? And this is the maternal. It originally came from the egg. Okay, so we're going to do blue to represent the paternal, red to represent the maternal. Okay, this is a homologous chromosome. They are the same size, they are the same shape, they have genes for the same traits at the same place, although they might have different alleles. Okay, then those chromosomes are replicated during DNA replication during the S phase, and now you have these homologous chromosomes exist as sister chromatids. Okay, so this is something I expect you to be able to do. Is if given something like this, you should be able to circle sister chromatids, right? So these are the sister chromatids. Okay, and you would get the same thing over here in the maternal chromosome. Okay. Sister chromatids are identical to each other, so if there's a big A on one, there'll be a big A on the other. If there's a little A on one, there'll be a little A on the other. Okay, but then this is also a homologous pair, right? So I'll circle in green the homologous pair. So that's a homologous pair, or homologous chromosomes, or homologs. Any of those words will do. All right, so I put this in just so that we could remember what's what, okay? So if I told you to circle the homologous or the sister chromatids or a pair of sister chromatids, okay, sister chromatids, I'm going to outline them all in this dark blue. Sister chromatids. So each chromosome consists of 2 identical sister chromatids. And find the last one, okay? And then now we want to find the homologous chromosomes. Homologous chromosomes, don't get confused, are not the 2 blue ones and the 2 red ones. Instead, they are the 2 of the same size. Okay, so these are homologous chromosomes. Pretend that little tail end is included. So the 2 big ones are, and the 2 little ones are homologous chromosomes. Okay, so in this case, this cell has 2 types of chromosomes, and it has 2 of each of those. All right, which leads us to the term diploid. Diploid is the state of having 2 complete sets of homologous chromosomes. In other words, every chromosome has a pair, and you have 2 copies of every chromosome. And because you have 2 copies of every chromosome, you have 2 copies of every gene. We can shorthand a diploid 2n, where n is like number of chromosomes, and you have 2 of those, so each chromosome. Okay, all diploid cells have homologous chromosomes, so each chromosome is going to have a buddy. It's going to have its homologue. Okay, human cells, our regular adult cells, are diploid. And here's back to this example. So this is a diploid cell. There are 2 types of chromosome, the long one and the short one, and you have 2 of each. So remember that, say if this was a fertilized egg, you would have the sperm that donated the blue ones and the egg that donated the red ones. Okay, so 2 types of chromosome, 2 of each. That's diploid. And contrast, well, okay. I was going to contrast that with haploid, but first I wanted to talk to you about the human karyotype. So a karyotype is a snapshot of all of the DNA in a cell. It's made by taking an actual cell that is going through mitosis and already has replicated DNA, so it's really hard to tell on these, but these are actually composed of sister chromatids, not a single chromosome. Each one is replicated. Okay. And what people do who are trained in this is they can stain the chromosomes, and then they line them up with their buddy. Okay, so they find the homologous pairs. Humans have 23 pairs of homologous chromosomes, and they are numbered. So we have 2 of chromosome number 1, 2 of chromosome number 2, 2 of chromosome number 3, etc. And the only thing that doesn't follow that pattern is right here when we get to the X and Y. The X and Y chromosomes are the sex-determining chromosomes. Someone who is XY is male. Someone who is XX is female. We'll talk about that more when we get to genetics. Just know that the X and Y, even though they don't look the same, they're actually pairs. They're homologous pairs. Okay, so humans have 23 pairs of homologous chromosomes, so that means we have 23 types of chromosomes, and we have 2 of each of them, meaning we have 46 total chromosomes in each of our cells. We have those same 46 chromosomes. Okay, now I get to contrast it with haploid. So haploid is a state of having just 1 set of chromosomes, and so that is N. There's no 2 involved. Okay, there are no homologous chromosome pairs when we talk about haploid cells, because only diploid cells have homologous pairs, because you have 2 of each. So we just have just 1 of each. Okay, human gametes, or the eggs and the sperm, are haploid. Those are the only cells in humans that are haploid. Okay, so here's an example. I took away the maternal chromosomes and left only the paternal chromosomes. So this could be an example of what kind of cell? That's right, a sperm cell. Except a sperm cell would actually have very little cytoplasm, basically just the nucleus, and it would also have a tail on it for swimming. Oh, and here is a lovely typo. So in this example, N equals 2, so there's 2 types of chromosome, but are there 2 of each type? No, wrong. Got to fix that. There's only 1 of each type of chromosome, and that's why it's haploid. So there's 1 long and 1 short. They don't have their buddies. So here's a karyotype for an egg cell. So here you can more clearly see that each chromosome consists of 2 sister chromatids, and then you only have 1 of each type of chromosome, and the egg is carrying an X chromosome. Here's one for a sperm, same thing, except for it's carrying a Y. Human sperm cells could actually carry an X or a Y. There's a 50-50 chance. And we'll talk more about that in genetics. All right, we're ready to go through meiosis. So we're going to go through the steps of meiosis, but first a little overview of meiosis and why we need it. Okay, so you're going to start meiosis with a diploid parent cell. Remember, meiosis takes place only in specialized cells that are either in the ovaries of females or in the testes of males. It's not just any old cell that gets to go through meiosis, very special cells. Okay, but the starting cells are diploid. So for a human, it would be 2n equals 46 for our starting cells. That would be way too many chromosomes to look at for an example, so we'll just reduce it and we'll pretend the lovely organism just has 2 types of chromosome. Okay, at the end of meiosis, we are going to end up with 4 haploid daughter cells. So in this case, they'll be n equals 2. In humans, it would be n equals 23. So each egg or sperm cell is going to carry 23 chromosomes, or half the chromosomes that are needed. Okay, so remember, these are the eggs or sperm that are used in sexual reproduction. So one good question might be, why the heck do gametes need to be haploid? Well, it's really important to maintain a constant chromosome number across generations. So organisms are made so that they will function properly when they have their right number of chromosomes. Having extra chromosomes can lead to malfunction and can lead to cell death. It can lead to, death of a fetus, for example. So you want to maintain the constant number of chromosomes across generations, and this happens when you, during the process of meiosis, you cut the number of chromosomes in half, say for a human, so you'd have 23 chromosomes in the sperm, 23 in the male. Don't want that there. Okay, 23. And then during fertilization, when the egg and sperm come together, you'd end with the diploid zygote, or 46. That's then going to go through mitosis and make an adult, right? Going back to that slide way back at the beginning, right, when I talked about what mitosis does. Okay, so you take the fertilized egg or the zygote and go through mitosis. So you want to have back to that 46 chromosome number. So having meiosis that reduces the chromosome number in the sex cells by half then allows us to get back to the 46 in the adult, and that's happy, right? That's the way it's supposed to be. What happened if you didn't? What if say a diploid organism made sperm by mitosis? Then you would end up with a diploid sperm. So in the case of humans, that would be 46 and 46. And let's see if I can do my math right. In the organism, that would, or the zygote, you would then have 92 chromosomes, right? And then if that same thing kept happening over and over again, you would increase the number of chromosomes exponentially. So you'd be doubling it every generation, and suddenly there'd be way, way, way too many chromosomes, and that just wouldn't work. Okay, so that's bad, right? So we need to have meiosis in order to have the sexual reproductive life cycle, so that we can recombine DNA with another organism and create novel combinations. All right, now we get to go through the process of meiosis. So there's 2 stages. There's meiosis 1, and in that the diploid parent cell divides and creates 2 haploid daughter cells. So this is where a diploid cell becomes haploid, is in meiosis 1, and then and in order for that to happen, homologous chromosomes are separated into separate cells. But each chromosome still consists of 2 sister chromatids. Then there's meiosis 2, and in that case, the 2 cells that you end up with here at the 2 daughter cells are going to continue through meiosis 2, and they're just going to separate out the sister chromatids into separate cells, and in the end, you'll end up with those 4. Once you get the process of mitosis down, and then when you start studying meiosis, you can look to see how meiosis 2 and mitosis are similar to each other. All right, we have the same names. Is that confusing or not confusing? I don't know which. It could be either. For meiosis 1, we just stick a 1 behind each one of them, and for meiosis 2, we put a 2 behind each one of them. All right, again, here's a video. Make sure you watch this. It's a great video. Goes through the process of meiosis. I recommend watching it now. Then I go through the process of meiosis in the lecture. Then you watch it again. That's what I suggest. Okay, so meiosis. Meiosis 1. So meiosis 1 is going to start out with prophase. Okay, in prophase, chromosomes will condense, just like in prophase of mitosis. But very importantly, homologous chromosomes will pair. So they find their other homologous chromosome buddy, and they pair up. It's called synapsis, and they form what's called a tetrad. That tetrad is now linked together by proteins, and it hangs out as a unit on its own, and they move together as if they were one chromosome. Okay, the nuclear envelope is always also going to break down. Okay, and the mitotic spindle fibers will attach to the centromere. Okay, one thing I don't talk about much in this lecture, but your book talks about is crossing over. Ooh, sorry, just trying to get rid of that. Crossing over. Crossing over is the process of exchanging DNA between the tips of the maternal and paternal homologous chromosomes. Increases genetic diversity as well. Okay, anyways, then you get these spindle fibers attaching to the centromere. It's going to move each pair of homologous chromosomes as a unit. Okay. In metaphase 1, the homologous chromosomes, or the tetrads, line up at the metaphase plate. And this diagram is a little misleading because it's showing them already pulled apart, when really right now at metaphase 1 they would be attached. Okay. Then in anaphase 1, the homologous chromosomes are separated. Okay, so if you learn one thing about meiosis 1 is that the homologous chromosomes are separated, right? And then they're going to move towards the poles. When they reach the poles, you're in telophase 1. Now each cell is haploid. Okay, and cytokinesis is also happening, so they're forming two cells. So each one are haploid. Back here we had 2 long ones and 2 short ones. Now we have 1 long one and 1 short one for each. Okay. The cytoplasm will divide and we'll end up with 2 haploid daughter cells. Okay. Each one of these daughter cells is going to go to meiosis 2. Okay, and that's what comes next. Okay, meiosis 2 starts with prophase. So I should say, often in textbooks it talks about the nuclear envelope reforming and cytokinesis happens, and you get these 2 haploid daughter cells. In a lot of organisms, and especially in the males of the organisms, the process of meiosis is happening so quickly that there's not really time for a nuclear envelope to reform around the nucleus before the cell goes directly into meiosis 2. Okay. So if the nuclear envelope reformed after meiosis 1, then it will then be taken apart again in meiosis 2, so you can see it wouldn't be very logical to put it together in the first place. Okay. Prophase 2 is just like prophase of mitosis, except the cells are haploid instead. So spindle fibers are going to attach. The chromosomes presumably are already condensed, because just like the nuclear envelope, they're not going to uncondense, just have to recondense again. Then the cell goes into metaphase 2, where the chromosomes align at the metaphase plate again. Now we just have this is exactly like in mitosis, right? So a sister chromatid from each chromosome is on one side or the other. And then anaphase 2, okay, here, this is where sister chromatids are separated. So again, if you learn anything about meiosis 2, it's that in anaphase 2, the sister chromatids are separated, and they're going to move toward the poles, and then those are going to they'll go through cytokinesis, and each one will create 2 daughter cells. Okay, they are already haploid, but what we have done here is just separate the sister chromatids from each other, right? So no more replicated chromosomes. At the end, we have these 4 haploid daughter cells. Okay, they are different from each other because of crossing over and because of how the maternal and paternal chromosomes align, and they are definitely different from the beginning cell, the parent cell. It's good to kind of just stop and pause and contemplate the difference between meiosis and mitosis. Okay, so in mitosis, we generate 2 daughter cells, which are diploid, genetically identical to each other, and genetically identical to the parent cell. And I just really like this cute little guy here. Here's a diploid little guy. Chromosomes lips, eyes are all duplicated. Those are supposed to be the two copies of the same chromosomes. Then they're replicated, then they're divided, then you end up with two identical to the first one. Okay. Contrast that to meiosis, where at the end you get 4 daughter cells that are haploid. They are genetically different from each other, and they're genetically different from the parent cell. Okay. Now we get all this division, and here we get haploid. All right, so now you all know about cell division, and you know what they're used for, but why the heck should you care? Well, understanding cell division allows us to understand things like cancer and Down syndrome as just 2 examples. So they do have definite application aside from just understanding how your own body works and those of other organisms, including plants and seaweed and fungus and bacteria and all that stuff, right? Okay, first, we're just going to be real brief about this, but first of all, cancer is loss of cell cycle control in mitosis. So cells go through uncontrolled mitosis, basically dividing when they're not supposed to. That happens when there are multiple mutations in a cell in the genes that control the cell cycle. So those stops that are in G2 that are, questions to the cell like, oh, did I do these things correctly? No more. Those regulations are not in place anymore, and the cell doesn't have to get any signals from the surrounding tissues or anything to divide. It just divides on its own, so it divides out of control, and that forms a tumor. Then when it's really bad, it's when the cancer cells start to metastasize, and so that's when they move out of just a single tumor. They move actually through the blood cells and can lodge in other organs and start growing more tumors there. And that, of course, is very bad. So you can think of cancer as a disease of the cell cycle. Okay, and then there's Down syndrome. Down syndrome is the condition when a person has 3 copies of chromosome 21. This, of course, is abnormal because everyone is supposed to have 2 of each. Okay, this is also called trisomy 13. Tri as in 3, somy as in number, or chromosomes. So did I say 13? Trisomy 21. And this is caused by a mistake during meiosis. Okay, so in normal meiosis, remember that in meiosis 1 here, that the homologous chromosomes separate. Okay, so if there's a mistake in meiosis 1, and instead of the homologous chromosomes separating, for one pair, it goes to the same pole, then you're going to end up with gametes that have the wrong number of chromosomes. Okay, so if it happens in anaphase 1, these 2 chromosomes are going to end up in one cell, and this one will end up by itself. So this, of course, we're talking here is a 2n equals 4 cell. Okay, in the end, these will still go through. So this is meiosis 2, this is meiosis 1. These cells will still finish meiosis 2, but the 2 resulting cells that came from this one that got the extra chromosome will have an extra chromosome, so it'll be n plus 1. So in this case, they would have 3 chromosomes when they're supposed to have 2, and the other ones are just going to end up missing 1 chromosome. Okay, these ones would be destroyed by the body. Those are not going to be gametes that'll last, but for some reason, these ones can go on and participate in fertilization. Okay, and so if you end up with a sperm or an egg that has 3 copies of that chromosome, or excuse me, 2 copies of that chromosome. Let me change colors here. Let's see if I can erase these. Okay, so that's the normal chromosome, and then here is 1 copy. That's what it should be, but this one got an extra green one too. Okay. So if that cell combines with a normal sperm cell, so we'll pick on the egg cell just because it most frequently happens with that. Okay, so we have 2 green ones here and 1 green one there. What we're going to end up with at the end, so we'll have fertilization. Let me label these egg and sperm. If fertilization happens and then ends up with a zygote, we're going to end up like normal with 2 blue ones, so that's right. But we will end up with 1, 2, 3 copies of the green one, and that right there is a trisomy. Okay, so that's bad news, right? Then you have 3 copies of that. So going back to here for a second, so trisomies can occur in any of the chromosomes, but usually we just see people living who have either trisomy 21, or you can also have trisomies of the sex chromosomes and live with hardly any effects, but maybe problems reproducing because you can't do meiosis correctly. Trisomies of all the other chromosomes lead to early miscarriages, lead to fatalities within, before the infant is born, just because their bodies are so messed up they can't survive. Chromosome 21 is quite small, so it doesn't carry that much genetic information, and that's one of the reasons why it's thought that people can survive having 3 copies. Okay, so on to this slide. The incidence of trisomy 21, or I would say trisomies in general, because you could also have the same curve for miscarriages within the first trimester, and those are often caused by trisomies for chromosomes which cannot be survivable. But as a woman's age increases, the possibility of having a child with Down syndrome increases dramatically after age 35. Okay, and that is because of how meiosis happens in a woman. So actually, during during embryogenesis, very early on after after fertilization and just when the embryo is just forming, that is actually when the egg cells start to be formed, and they enter into meiosis. They start the process of meiosis when the homologous chromosomes are paired, and then they pause, they stop, and they wait all the way up until puberty. With the onset of puberty, then one egg each month finishes that process of meiosis and is released as an egg. Okay. As a person gets older and older, those eggs have been sitting there for longer and longer with those homologous chromosomes held together and with the spindle fibers attached. And the reason why the increase of trisomies increases with age is just because the mechanisms for pulling those homologous chromosomes apart get old, right? So they just start malfunctioning, the proteins start breaking down. All right, that is the end of our cell division lecture. Thanks a lot for listening.